I'm Meredith. I'm a dietitian. Um, and so today I want to talk about the role of oxalate and other things like polyethylene glycol, vitamin C, glycine, glyphosate, and how they impair lactate dehydrogenase. And that results in low cellular levels of NAD, a cofactor that's needed for vitamin A metabolism. And I'm going to show how these particular substances can actually cause a vitamin A toxic state. So just a disclaimer, I'm a dietitian. I'm not a doctor or your family member's doctor. Please consult with your personal health care provider prior to making any changes in your diet, supplements, medications, or lifestyle. This video is just meant to inform. It's not meant to diagnose or treat. So these are different foods that have oxalate. This isn't a comprehensive list. Some of the foods are raspberries, chocolate, beans, turmeric, sweet potatoes, regular potatoes, stevia, nuts, spinach, carrots, beets. Pretty much most vegetables and fruit have oxalate in varying amounts. Some are low, some are high. And so it's the very high oxalate foods that are causing damage. And then we question, how do these foods get so high in oxalate? Were they always high in oxalate? Actually, I believe that the oxalate content of food has increased over years because of the introduction of glyphosate into the environment. So when vegetables are sprayed with glyphosate right before harvesting, we get that glyphosate and we eat it. So like some grains are sprayed with glyphosate to dry them out. When we eat that glyphosate, we absorb it and we metabolize it to oxalate in our body. Likewise, when it's in the groundwater, the plants suck it up and the plants will make oxalate inside of them as well. So I think that's where the massive amount of oxalate is coming from. And it explains why you can't use very old lists of foods, because sometimes they'll be considered low oxalate on a list that was made 40 years ago, but now it's a high oxalate food. So this actually happened with plantains. I was following a handout that said that plantains only had 1 milligram of oxalate per plantain, but in actuality, it's now up to 525 milligrams. So plantains, even though they're not on this list, they're definitely not a safe food for people to eat. And a plantain was actually what gave my 11-year-old oxalate poisoning. I was slowly giving it to her over many years, thinking it was low oxalate, and then it culminated in a severe poisoning with plantain flour, and she ended up in the emergency room. And that's when I made the connection to her current vitamin A toxic state. So another thing that can become oxalate in the body is actually vitamin C. Ascorbic acid is just degraded. It's not using an enzyme. It's just degraded into oxalate. And so then this can come out in the urine. Also, we'll talk about this in a little bit, how glycine plays a role. So I don't agree with taking vitamin C until bowel tolerance, because I actually believe what bowel tolerance is bowel death. So when oxalate levels are too high inside a mitochondria, the mitochondria actually won't be able to use NAD anymore. The NAD levels go way too low, and this ends up killing the cell. And so I think that the diarrhea that happens from excess vitamin C could potentially be actual cell death and the sloughing of the intestinal tract. So excess vitamin C isn't really that good, in my opinion. So polyethylene glycol also is metabolized to oxalate, and it actually impairs our ability to make the cofactor NAD in two ways. So one, I'll show you, it actually causes high levels of oxalic acid or oxalate, and that oxalate is going to impair the ability to recycle NAD, and then also ties up the enzymes that are used in vitamin A metabolism. So alcohol dehydrogenase and aldehyde dehydrogenase are used in vitamin A metabolism. So large intakes of polyethylene glycol actually can contribute to the problem of having a very high oxalate burden and also the problem of having these enzymes tied up. And we absorb 3.7% at every dose of polyethylene glycol. So we're told that it's not absorbed, but the studies that were done using polyethylene glycol and saying it was not absorbed, that's polyethylene glycol 4,000 or greater. And what is in most products containing polyethylene glycol is something less than 4,000, usually 3,500. So at least 3%, 3.7% of that is absorbed. So I was talking before about how glyphosate is a source of oxalate. Glyphosate is metabolized to oxalate in plants. Humans ingest glyphosate. We can make that into oxalate. And then the plants also will take glyphosate and turn it into oxalate. The glyphosate conversion in the body can also produce formaldehyde, which is also going to hurt our NAD levels because it requires NAD for processing as well, because aldehydes have to be processed through aldehyde dehydrogenase. And our water quality counts where we live. If you have hard water, then glyphosate stays in the water for years. If you have soft water, it starts to degrade within a couple of weeks. Most of our cities have hard water. That's because we clean the water and they have to use chlorine to clean it. And then they also typically add back in calcium and other minerals. And so that makes the water hard. Okay, so oftentimes people are concerned about glycine because glycine can actually enter the glyoxylate pathway and become oxalate. And it also uses up this NAD. So it makes a low NAD state when you overburden the body with glycine. So glycine is typically found in collagen peptides. You can actually supplement with glycine. I wouldn't suggest it. It is sweet, so sometimes it's used as a sweetener in products. But there's actually way more ways that we can get an excess amount of glyoxylate in our body besides just glycine. Actually, there was one study showing that hydroxyproline is metabolized more to glyoxylate and then oxalate than glycine itself. But again, hydroxyproline is going to be high in things like collagen peptides. So the center of this pathway is actually Glyoxyl. Okay, so if we go back, so we have the Glyoxyl right here, and that becomes glyoxylate. And so there's lots of ways that we can get Glyoxyl in our body. And that will be a different presentation, but I wanted to talk briefly about it. So we make some Glyoxyl in metabolism. We have glucose, and it can eventually come through these pathways and become Glyoxyl. We also take glucose and lysine and arginine, and we make a Schiff base, and then that becomes this product, a Midori product, and then that also becomes Glyoxyl. So if on a lab, so say that you do a plasma amino acid test and there's a large amount of lysine, that's a potential risk factor for making this Schiff space. And so it's something to consider. Maybe this particular person, if they have higher lysine or arginine levels, have a risk for making a Schiff base. Also, though, high lysine on an amino acid lab could be that there's a block in the kynurenine pathway. So maybe they're not making enough NAD in their body. And so also you can make this Glyoxyl out of ascorbate. And here's something really interesting. Excessive amounts of polyunsaturated fats, so seed oils, and those seed oils, or every product on the market has like canola oil or sunflower oil as the oil in it. Those actually go through lipid peroxidation, and they will become Glyoxyl and be a source of making oxalate in the body. But the major source that I've seen is actually dietary oxalate causing this problem. So when we eat excessive amounts of oxalate in our diet, oxalate, whether it's made in our body or whether it's eaten, it actually has a feedback mechanism because our bodies don't really like to have this poison laying around. It'll actually come back and stop this enzyme right here, which is called lactate dehydrogenase. It's the last enzyme in the production of oxalate from glyoxylate. So inside the body, just from our natural metabolism, this isn't a bad thing. But now that we have a large abundance of oxalate in our environment, in our diet, now we're seeing an unregulated intake of oxalate. And that ends up blocking this enzyme, lactate dehydrogenase. So that enzyme plays a role right here, but it also plays another role in metabolism. Lactate dehydrogenase is the major enzyme that drives pyruvate to lactate in the body. And so if this enzyme is blocked, then you cannot turn pyruvate into lactate. And so what you would see on a lab would be that lactate would be very, very low, or pyruvate could be extremely high, but there's an imbalance in them. For my daughter, her lactate was very, very low. And so that was shocking for me. And I realized that she was having a mitochondrial issue. And so when lactate is very, very low, then you also aren't getting a recycling of this. It's called NAD. It's a cofactor. And it's basically like an energy helper to move reactions forward. So her NAD was super duper low. And you could see this in her life. Like, she was very tired because another thing you use NAD for is to make ATP in the TCA cycle. She was exhausted, and she was also overweight, and she still is because it's taking her a while to get back, to normal metabolism. And so a symptom of low NAD is actually obesity. And so that's pretty interesting. So this NAD, though, is crucial. Vitamin A metabolism is dependent on having adequate cellular levels of NAD. So you're either in an NADH state or you're in an NAD state. And it's kind of like cycling back and forth. And oxalate basically stops the recycling of NAD and NADH. And there's actually many other things that can do this as well, which will be in other videos. So oxalate is a big one. So it decreases the cellular levels of NAD, which makes it so that alcohol dehydrogenase and aldehyde dehydrogenase cannot move forward. So what ends up happening here is that you get stuck in the retinol and retinal form of vitamin A. Now, we all know that retinol is used in the visual cycle, but you don't need a ton of it. If you have a lot of this in your body, it starts to cause problems. It actually can bind to an ether lipid called ethanolamine, and I call it ethanolamine steel. This actually happens in the eye. It's what leads to macular degeneration. It causes an A to E shift base. And I actually think this is what is causing low ethanolamine levels in people who have Alzheimer's disease and autism. They have low levels of ethanolamine on their labs. And I think that the retinal is stealing the ethanolamine and causing brain degeneration because it can't make ether lipids. Also, that stealing of, it's actually phosphatidylethylamine. It leads to a choline deficiency because you use phosphatidylethylamine to make choline in the body. Also, to be able to get the retinol out of the body, not out of the body, out of the liver, you have to have enough choline to transfer it onto retinol binding protein. So you're basically in a low choline state, and it's hard for you to mobilize this vitamin A from your liver. And at some point, you have maxed out your retinol binding protein, and so that'll be very, very high in the blood, and it will cause a high serum retinol level. So there will be another video about all the effects of high retinol and high retinal But when you're in this state, because you're low on NAD, you are not making retinoic acid. So what you'll actually look like, you'll look like you're vitamin A deficient. You'll have keratinization, you'll have keratosis pilaris. You actually will have problems with your vision. You'll probably lose hair, including the outer third part of your eyebrow. Um, you'll eventually develop other symptoms of having high vitamin A but low retinoic acid. And again, that'll be in a future video. So one more thing to talk about is that I had always learned that beta carotene was safe. But what I do know now is that it is not as safe as we thought. So beta carotene can enter at the retinal level. And from kinetic studies, we know that typically we do not convert retinol back into retinol. So we have this very, very high retinol state. And when you add too much beta carotene, it pushes this even further. And I think that it is contributing to brain degeneration in many people. So, yeah, it's devastating to know these things and also to see them happening in my own child. It's quite sad. So the good news is that I'm figuring out ways to help people, and have a really good group of scientists and people who have been through it who are contributing, and it's a wonderful process. So if you have any questions, please feel free to comment. My website is weaktherefore strong.com, and there's a lot of blogs about this as well.